Okay, before we get started, a couple of quick announcements. The first one is that those of you who are looking for the PowerPoint of the lecture notes last night, I had it done, of course, because it was Sunday, and it was done way too early for me to upload it, and so I spaced on uploading it till this morning. If you're looking for it, particularly early in the morning, please send me an email and remind me about that. I did get audio, thank you, for the abbreviated lecture, so that's been posted on YouTube as well. I've been really good about posting all the links to all of those in the recordings. I'll try and do that. The easiest way to see those recordings is just to subscribe to me on YouTube. I know it's, makes me feel wonderful, no, but it's just the easiest way to know when I have a new posting, which is there, which is 99% lectures. A few other things as well that are there. Another quick announcement, syllabus-wise, a couple of people have asked me about what's going to be on the exam. Is what's on the syllabus, what's going to be on the exam? Is it what you get through in lecture going to be on the exam? It's what I get through in the lecture. And those of you who did have a chance to look at lecture notes from today may have noticed that the genomes part is really, really short, if at all. So we may not end up talking about genomes for this midterm. I'm going to try and slide it in a little bit later if I can in terms of the course. And then the last thing, announcement-wise, has to do with this seminar today. In the physics department, of all places, one of the things that's happening at PSU, some of you may or may not know this, is we're trying to hire a whole bunch of people involved in environmental extremes and thinking about how life can survive, how people can survive, how we can get sustainability, et cetera. And that's through physics, chemistry, biology, environmental sciences, geography, English, communications, et cetera. And one of those people is Dr. Jay Nadal, currently at Caltech, who's, I don't like this title at all, Spying on Nature as It Moves, New Technologies and Probes for Cellular Imaging. She's developed these really amazing holographic microscopes, which just holographic microscope is just too cool anyway, just thinking about it. And particularly using this to apply to environmental biology, environmental microbiology. She also works closely with the Jet Propulsion Lab. In fact, I think she worked there for a little while beforehand. So these are sort of the microscopes we might put on robotic spaceships and go to other planets and look for life, which I think is really, really, really cool. So should be a really fun seminar. It's at 3:15 this afternoon over in 107 in Science Building 1. So any questions about sort of, exams and so on and so forth, how the class is running so far? Good. So we'll move on here. Oh, the other thing was, well, we'll get to it just in a second. Clicker questions. I have uploaded clicker scores. Hopefully people saw those on D2L. If for some reason your scores are not showing up on D2L and you have a clicker and you are using it, make sure you've registered on iClicker.com, and if that's still not working after about a week or so, send me an email with your clicker number and I'll try and update it by hand. Most of you seem to be in there, which is nice. So that's looking pretty good. Speaking of clickers, after all, let's get started with that. How many protein subunits are in a nucleosome when it's a bead in the beads on a string chromatin state? 1, 2, 4, 6, or 8? Ten, 5, so we seem to have a bit of a difference of opinion here. Basically between 4 and 8. There are a couple of key things about this particular question. The first one is how many protein subunits. When we talk about subunits, these are the number of individual polypeptides that fit into a particular structure. It doesn't say how many different proteins are involved. If it were the number of different proteins that were involved, then the answer would be what? 4, because it's H2A, H2B, H3, and H4. However, that's not what the question asks. It's how many subunits are there. So what does that mean? 8, correct. So that being said, we're going to continue talking about chromosome structure today, mostly in terms of what comes after that nucleosome formation. But I wanted to talk a little bit about nucleosome assembly. This is actually not stuff which is directly in your textbook, but I think it's really important in terms of understanding the dynamics of nucleosomes. One of the things that we found out recently is that nucleosomes are much, much more dynamic than we ever thought that they were. And we'll talk about some of that, and a lot of it has to do with how nucleosomes come together as well. Histone modification. This is, everyone's heard about epigenetics? Yes? No? One of the main kinds of epigenetic inheritance. It's by no means the only one, but when most people talk about epigenetics, they're talking about histone modifications. We'll talk a little bit about heterochromatin. Heterochromatin is just compact chromatin. And then finally a little bit about metaphase chromosomes. So wanted to start out with this as something we should have really talked about last time as well, as how people really discovered nucleosomes in the first place. Basically what they did is they took DNA like this and they chopped it up with a nuclease. In this case, micrococcal nuclease, which is shown over here, MNase. And they used very small amounts of that nuclease for short periods of time, and what they found was they had pieces of DNA that were mostly about 140 nucleotides in length, but some that were about 280, etc. And this is what they call the whole ladder structure of these individual nucleosomes, which says if you take the micrococcal nuclease, nucleases, enzymes, they chop up nucleotides, and so they can cut, but they can only cut where the DNA is not protected. And it turns out micrococcal nuclease also cuts really nicely in the minor groove of DNA, and that's where a lot of the nucleosomes are interacting. So this is how people first found this. And then what they also did is they purified the proteins that were associated with each of these individual nucleosomes, and that's where H2A, H2B, H3, and H4 come from. And in fact, this image here is from a paper that I found published a couple of years ago, where people were literally looking at the proteins that were present associated with each of these individual nucleosomes. So just to show on their paper, they showed that, yes, we've got good nucleosome preps in this process. So how do you make nucleosomes? Let me move this thing out of the way. Sorry about that. No, if I can. Let's see. This is, well, nucleosome assembly. It's behind that multiple choice thing there. Basically, nucleosomes get put together first by H3 and H4 coming together, and H3 and H4 form what's called a heterodimer. We haven't talked about heterodimers too much yet. Heterodimer just means two different proteins that come together. One is a monomer, two coming together as a dimer, and then those dimers come together, which I saw shown here as the H3, H4, 2. So you have two dimers that come together. That makes a tetramer, so 4. And then that associates with the DNA. And so that's what you see here. Starts to wrap the DNA around it. And then you have H2A, H2B, which also form heterodimers. And then those get added here to this forming nucleosome process. And so this reason that I go into this really has to do with when you think about alternative nucleosomes, that we'll talk about in just a second. Think about the ones that are most likely to be exchanged. It's going to be these guys which come in at the end, which are the ones that are most likely to be exchanged. So if you think about alternative nucleosomes, those are the ones with the exchanged histones that come in there. To do this, you need extra proteins called chaperones. I like to think of chaperones, given that I have 2 young daughters, as trying to make sure that the wrong interactions don't happen and help the right interactions happen. So that's what's going on here. But you really need these extra proteins in order to get this process to take place, and particularly these exchanging of the individual subunits that are present there. Each of the subunits that you have in your nucleosome, they're all histones. Each of these histones is a very similar structure. That structure is pretty basically one long alpha helix with two other alpha helices that cross over it. And then that forms a dimer down here. That's shown for H2A, H2B here, but the very, very similar structure happens with H3 and H4 as well. So that's what's called the histone fold, is this helix with two other helix on top of it, which then are going to interact with each other and then eventually interact with the DNA. A couple of important things to look at here. All of the histones are very similar in terms of their structure and also, not surprisingly, in their sequence in this region where they have this same structure, but they're very different, either N-terminal to this structure or C-terminal to this structure. These are what people call tails. So the histone tail, people talk about a whole bunch: N-terminal tail, C-terminal tail, and we're going to spend most of our time talking about the histone H3 tail. These tails stick out of the nucleosome on the outside, and that's where most of the modifications happen, again, that we'll get to in just a second. Last time we talked a little bit about how the DNA is wrapped around these nucleosomes. I've got my sort of nucleosomal DNA here. It's not quite right, only 2 loops rather than 4. But the proteins that are present on the inside of this, you've got to wrap the DNA. And clearly it's not like the DNA that Watson and Crick came up with, which is this nice, actually pretty straight process. And so to be able to bend the DNA, it's actually pretty hard to bend the DNA in such a way to wrap it around each of those nucleosomes. And it turns out that ATs are better in the minor groove of DNA to be able to squish them together when you're wrapping things around. And because of that, since it's every 10 base pairs between here and here, that was that big repeat that Watson and Crick saw in Rosalind Franklin's X-ray image, that every 10 nucleotides, if you have ATs, then nucleosomes like to form around those particular sequences. And they form there slightly better than around other sequences. True, it happens with all sequences, but A's and T's spread every 10 nucleotides a little bit better at forming nucleosomes. So you've wrapped your DNA around your nucleosome. Again, this is a couple extra strands here. One of the big problems that happens once you've wrapped it, you've bent it pretty seriously, but then where are all of the sequence-specific interactions going to happen? In the major groove of DNA. Now, access to the major groove of DNA, if you're wrapped around, yes, it likes to bind to the minor groove, but there's certainly going to be major groove as well that's associated. And here's a nice example. Excuse me. My pointer is giving up on here. Not quite sure why that is. But here you see it's a nice major groove. If the site, the DNA sequence that you want to bind to is sitting right here, how are you going to get a protein interacting with this? Well, it's not going to happen. And so this gets back to the whole idea that nucleosomes are not really stable structures. Even though wrapped around, people always used to think of these, nice, stable protein DNA interactions. It turns out that the DNA-protein interactions here are very flexible. They dissociate, reassociate, and this is a process that's also called nucleosome breathing. And that basically says that the DNA that's wrapped around the nucleosome comes off, goes back on, comes off, goes back on. And because of that, you can have interactions of DNA binding proteins to sequences that are otherwise bound in nucleosomes just by letting them sit there at 37 degrees, because these things are flopping around all the time. So nucleosome breathing is one way to get access to sequences which are there. Turns out that that's not the only way, and if it were the only way, probably lots of things wouldn't happen or take a lot longer for things to happen. So there's a whole set of proteins called chromatin remodeling complexes. These chromatin remodeling complexes are ATP-dependent complexes. Complex means it's got multiple proteins that are involved in that. And that ATP hydrolysis seems to allow the DNA to slip around the nucleosome. It's a little bit hard to see here, but what they're trying to show is that with this little blue piece right here, this is where it starts when you have your chromatin remodeling complex. You have ATP hydrolysis, and this sequence moves around the nucleosome to end up here. And in that process, this piece ends up over here, so this particular DNA sequence. It's really unclear how this happens, how you take ATP hydrolysis and then basically sort of rotate the nucleosome relative to the DNA. But potentially, breakthrough happened literally 2 weeks ago in Science magazine. This is citation from 2017 here. There's a group that was looking at how accessible the protein side chains are in the nucleosome when you have one of these chromatin remodeling complexes around. So a little bit hard to see here, but it's this blue thing over here. This is your chromatin remodeling complex. Basically what seems to happen is when there's ATP hydrolysis, this guy kicks the H2A and H3 molecules that are inside the nucleosome and basically makes everything wobble around. And so that's what all these wobbling is for. And in that process, the DNA can actually move. And so it's this wobbling of the internal complex, which seems to be the most important for allowing this rotation to take place, and that's the ATP hydrolysis that basically tries to get everything moving in that whole structure. So stay tuned. Next version of the textbook will probably have something kind of like this actually in there. So yes, molecular biology does move pretty quickly, and again, anything that's in the textbook is almost by definition out of date already. So that's one process that you have with these ATP-dependent chromatin remodeling complexes. That's just shifting the DNA around. These also seem to be very important for getting in some of those alternative histones that I told you about, H2A, H2B in particular, because those are the last ones that come into the complex. Those are much easier to replace than H3 and H4. These histone chaperones, sort of this blue piece here, also have ATP hydrolysis activity that seems to be important, probably also just like what we just saw for those chromatin remodeling complexes for bumping the pieces out in the middle. You can also have complete replacement of the nucleosomes. This happens more rarely than this exchange of individual ones right here. That's all I wanted to talk about in terms of actual nucleosome structure. Now let's talk about a little bit bigger picture, which is what really most of your DNA looks like most of the time. So there's also Histone H1, which was found earlier than the other ones, which is why it's called H1, versus H2A, H2B, H3, and H4. There are lots of different kinds of Histone H1. Histone, the core histones, so H3, H4, H2A, and H2B, are extremely similar to each other in sequence. There's only 1 or 2 amino acid changes in all eukaryotes, and even archaea. Some archaea have these histone-like proteins that are associated with them, so it's extremely well conserved. Histone H1s are much less well conserved. Those seem to sit on the outside of the histones and kind of help to hold those histones together. There are the tails, remember the N-terminus and C-terminal tails, which get modified but are also important for bringing these nucleosomes together. And most of that gets you to something called the 30 nanometer filament, which is basically what you can see down here. It's about 30 nanometers across from this side to that side. Is an electron micrograph of purified DNA from cells. And then there are lots of other proteins as well that are associated with this. Somebody asked me last time about, is it chromatin just the nucleosomes, the histones, and the DNA? No, there are a lot of non-histone proteins that are associated with DNA, all of these interesting little cartoons here as well. So we don't know exactly what the 30 nanometer filament looks like. There are a number of different models for how it fits together. A lot of hand waving. There actually are some high resolution structures for 2 nucleosomes together, and actually there's some for 4 together at this point, but much more than that, we really don't have a very good idea, and that's probably just because they're flopping around. Not all histones associate with each other in the same way. Last time I talked about how these histones are basically like disks, and so they can stack up against each other, and that stacking really does seem to be very important for how all of these are functioning. How do you get them to stack with each other? One way is through these tails that are hanging out, and these tails actually then interact with each other. So one tail from one nucleosome will be interacting with the next nucleosome along, and so on and so forth through the process. Now, talked about histone modifications. Those can be modified on the tails. If you have modification of a tail structure, that will probably also have change in your structure and change the interactions that it's interacting with. So it should be pretty obvious that any kind of change in that tail is also going to change how these nucleosomes are set up relative to each other. Here are a couple more transmission electron micrographs of this 30 nanometer filament. So here lined up what they call the zigzag fashion. But the whole idea here is basically that all of these nucleosomes sort of line up flat with each other. And it's a lot easier to see, I think, in a, excuse me, I've got my pointer here, I can move this out of the way. Looking in a video form, and that's here. This is linked. So here's a molecularly really pretty good example of what we think DNA looks like. First you have assembly of H3 and H4. Then you have H2A, H2B. Those then finally assemble into your individual nucleosome. Here, nucleosome. There's some commentary that goes with this that I'm not that wild about. And then all these nucleosomes start to associate with each other. You can hopefully see the tails flopping around on the outside here. Those are then assembling. This is a model. But another thing that I really like about this is that you see it's wobbling all over the place all the time. And that gives you a good idea of the dynamics which are going on in this whole process. The 30 nanometer filaments then all line up with each other, and those get assembled and eventually into a whole metaphase chromosome. And so we talked about the compaction that you have to have in terms of chromatin. Nucleosomes are only the very first step. Then you're having the assembly of the 30 nanometer filament, and then all of those come together, and eventually you have a metaphase chromosome. So let's go back to here. So another clicker question. Turn this thing back on, that would help. Which of the following requires ATP hydrolysis? Let's start. Nucleosome formation, nucleosome breathing, chromatin remodeling complex activity, and when Stedman can't come up with any other answers, he throws in these A's and C's or A's and B's. Ten, 5, 3, 2, 1, done. Just because everyone thinks it's right doesn't necessarily mean that it is. But in this case, yes, you're correct. Yes, it's the chromatin remodeling complexes. Nucleosome formation does not require any ATP hydrolysis, unless you want to really get nitpicky and talk about exactly how the proteins get formed in the first place. But I think that in this case, the actual formation of the nucleosome will be these things coming together. If a question like this comes up on an exam, you can go, Dr. Steadman, I'm not quite clear about what you mean about this. And I will try and do my best to clarify that. One thing about clicker questions is that they are not as well edited and proofread as the questions on exams, at least hopefully that will be the case. So nucleosome breathing is what happens. Again, you saw it in the video. And then chromatin remodeling complexes are definitely ATP-dependent processes. So let's look a little bit at histone H1. Histone H1, we actually do have a pretty good idea what the structure looks like of that. Here's an example right here of a nucleosome plus particular histone H1. It's actually missing a couple of the tails. These histones also have tail structures that also get modified. Binds asymmetric to the nucleosome, and the main thing that we can see with these H1s is they do seem to help get that 30 nanometer filament to form, but the easiest thing to say is if you do the experiment like I talked about right at the beginning, where you purify DNA, use a nuclease to cut it into smaller pieces, then about 20 more base pairs are actually protected. So instead of seeing a band at about 150 nucleotides, you see a band at about 170 nucleotides. And that's just because the binding of this protein here stops the micrococcal nuclease coming in and chopping up this particular piece of DNA. So we've got our histone tails, we've got histone H1. Now, what do we put everything else together? Again, you saw in this video that the 30 nanometer filament gets wrapped into these bigger structures, about 300 nanometers. These are what are called loop domains. This is something that you can see in the electron microscope if you purify the DNA from pretty much any cell. These are pieces of chromosome that are usually about 300 nanometers in length. If you just look at human chromosomes and regular cells, not those that are compacted and undergoing mitosis, you see about 2,000 of these. And what we know is that in those particular loops, because we know the sequence of the DNA, we can design oligonucleotides, small pieces of DNA with radioactive probes from the old days. Now people use fluorescent labels and see that those fluorescent labels will interact with that one particular piece of DNA. So what you can see is that these about 2,000 loop structures are where most of the protein coding genes are in each of those particular loop structures. And they're all separated from each other, each of those separate loops. And we'll see that in a little bit more detail. How do you get that separation? There's a lot of proteins that hold these loops together. And this is what's called DNA scaffolding. So we'll see, yes, the DNA itself and the nucleosomes are pretty dynamic, but they all get held in particular parts of the nucleus. And what does that holding are all of these so-called scaffolding proteins. Scaffolding proteins are also known as the nuclear matrix. So it's all the proteins that are left once you get rid of the DNA in the nucleus. And again, the molecular biologists are incredibly creative in terms of their naming. The DNA sequences that attach to the matrix are called the matrix attachment regions. But they are specific DNA sequences that are going to associate with things that are present in the nucleus holding particular parts of the chromosomes in particular parts of the nucleus. So we'll talk about some of those, but first I wanted to talk a little bit about histone modifications, because this is really where we understand the most about variations which are happening, and it turns out that these histone modifications are really critical for turning on genes and turning off genes. So mostly so far we've talked about the beads in the string, the individual nucleosomes, that actual bead itself, that's your nucleosome, so it's got the core histones, but they're not identical to each other by any stretch of the imagination. So it's that variation between the individual nucleosomes. Yes, they have the same proteins, but those proteins are all modified in different ways for different nucleosomes. And it turns out that's basically important for everything that the DNA in a eukaryotic nucleus does. So turning on genes, packaging your genome, segregating genomes, etc. So let's take a look at that. We know quite a bit about these individual histone tail modifications. Let's see if I can get my pointer back here. No, it's deciding to give up on me. Histone tail modifications. These are just some of the modifications that can take place. You're never going to have all of these modifications happening at the same time. This is a cartoon version of histone H2A, has an N-terminal tail, H2B N-terminal tail, H3 and H4, etc. Important amino acids here. Lysines, we're going to keep beating on lysines again and again and again here. Why lysines? Charged, yes, is one aspect of it. But why do they have charges? Because they have amino groups hanging out on the outside. We talked about the amino groups as being important for ubiquitin to be bound to it through these isopeptide bonds. And in fact, one of the modifications that's not shown on this slide is ubiquitination. Ubiquitination was actually first found in looking at histone modifications to a lysine in one of the histones. So the other thing that happens with lysines is you can have acetylation of up to 3 acetylations that can happen, up to 3 methylations that can happen. So these are the most common that are happening on these side chains, but there are other ones as well. Acetylation, which are the A's, methylation, which are the M's, and then P's, which are for phosphorylation. Now, phosphorylation happen on lysines or not? No. Where does phosphorylation happen? Side chains that have OHs on them. Serines, threonines, and what's the third one? So methionine doesn't have an OH at the end. Tyrosine. So we talked about kinase. We'll talk more about kinases later on. Protein kinases that are present in eukaryotic cells are going to interact with serines, threonines, and tyrosines. So serines and threonines, S and T. I don't see any T's actually on this slide, modification-wise, but certainly serines. So all these serines which are getting phosphorylated. What happens when you put on a phosphate group? Why is phosphate such a wonderful thing to be using for protein modification? It's early on Monday morning, I know. We haven't caffeinated enough. What's happening with phosphates? Why phosphate? Who cares? Phosphates have what? Negative charge. So negative charges you introduce in a histone. What are histones associated with? DNA. So negative charge you put next to DNA. What's going to happen? Going to push the things apart relative to each other. What about lysines? What kind of charge do they have? Positive charge. What are they going to want to do? Interact with DNA. When you modify your lysines, what happens? You're going to get rid of those charges, particularly if you think about the methylation that takes place. So these are some of the interactions here. The other thing that happens when you have these modifications on these side chains, which are sticking out relative to the nucleosome, particularly here, histone H3, any kind of modification that happens here is now going to either allow or block the interaction of other proteins that are going to be associating with the nucleosome complex. And so a lot of people talk about these modifications not as a direct kind of interaction with the nucleosome, but also in terms of interactions with other proteins. Yeah?
So with the different changes that the tail can make, so when the tails interact with each other, when they start combining, do the other tail also need to have a modification in order to accept the other modification?
So the question, I'm just going to repeat it so it's on tape here, and correct me if I'm wrong, is do you basically have to have symmetrical modifications? So say, for instance, H2A has the phosphate at the first position, at serine 1. Does the H2A in the next nucleosome in the 30 nanometer filament also have to have that phosphorylation? The answer is no, they're not symmetrical. So you're not always having just two coming together. It's a whole stretch of different ones. So it turns out that the tail of one is not always going to interact with exactly the same tail on the next one. But there are potentially reciprocal kinds of changes that you'll see. So a change in one will interact with a different change on another one. So again, these are a bunch of different possible kinds of changes that can happen here. And as I mentioned, most of these seem to be involved in not so much direct interaction with the DNA, although there's certainly some of that, but much more so in terms of interactions with other proteins. And these other proteins are called code readers. Why code readers? Because people talk about the modifications that happen on the amino acids in each of these histones to be a histone code. And we'll look at the histone code in just a second. We don't completely understand the histone code. So modification XYZ means A, modification PDQ means Y, etc. So, but the idea here is that if you have these modifications, which are shown as these little blue balls at the end of each of these histones, these are all different. So you could have, phosphorylation on the serine, you could have methylation on the lysine, you could have acetylation on a different lysine. All of these are going to be giving you a particular structure which can now interact with one of these code readers. Multiple different, here they call them modules, usually it's an individual protein, that's going to interact with some modification. And the classic one, in fact probably the best studied, is trimethylation of lysines, in particular lysine 9 in histone H3. We'll see why that is in the histone code in just a second here. So these code readers are going to have all these proteins going to interact with different modifications on different histones. If you have the appropriate kinds of modifications on the histones, now your code reader complex will bind to this particular piece of chromatin. If it does bind, then it will interact with other proteins. So proteins interacting with proteins. This is something that we'll see a lot. First you have proteins interacting with DNA. We had our nucleosomes interacting with the DNA. And then other proteins that interact with those, and then other proteins that interact with those. Sort of your whole Russian doll complex. But the idea here is that the protein that interacts with the histone code reader, in and of itself doesn't have any actual activity. But it can interact with proteins that do. And when I say activity, I usually mean enzymatic activity. And so that's just shown here as this blob. If we look at a slightly higher resolution version of this, no longer cartoons, basically the main message here is that these reader, and also we'll talk about writer complexes in just a second here, are way bigger than the nucleosome itself. These are really big complexes with multiple different proteins that are associated with the nucleosome complex. And those of you who've looked at the notes from last year, I had another example where it was about 5 times this big. So they're big proteins that are interacting with each of these nucleosome complexes. So what are they reading? Well, they're reading some modifications. Up the top here, this is just basically taken from that previous slide that we looked at. Slide. What do people call them slides anymore? As a slide projector. Yeah, sorry.
So this scaffolding protein in this complex, does it play kind of a similar role to the E3 and ubiquitin, it's called ubiquitin ligase, kind of just holding the other ones in the correct position?
Yeah, so the question here has to do with the terminology, so scaffolding, and is the analogy with the E3 protein in ubiquitin ligases or the ubiquitination complex, E1, E2, and E3. That particular culin, in fact, is usually the scaffolding protein itself. That's part of E3. There are other proteins that are involved too. That scaffolding protein is very much analogous. So it's a protein that holds a bunch of other domains in the appropriate kind of orientation to have activity. So you're exactly right, yes. That terminology is very, very similar. So, so this is the histone H3. These are all the possible modifications, and actually these are all modifications that have been detected in histone H3s that have been purified from cells, acetylation, methylation at different lysines, serines that have phosphorylation, arginines that get methylated, etc. And again, The one that's probably been the best studied is this one here, K9. It's not a dog. It's lysine 9 in histone H3 that gets methylated. Trimethylated state is connected to formation of heterochromatin. We'll look at that in just a second. Heterochromatin is the compaction of chromatin, making it really compacted. Once you've compacted it, then the other proteins can't get to it. And probably most important as far as we're concerned, the proteins that can't get there are the DNA-dependent RNA polymerases, which are necessary for making the messenger RNA, which is eventually going to make a protein. So if you have heterochromatin formation, it's going to shut down gene production. On the other hand, acetylation at K9 together with methylation at lysine 4 actually leads to gene expression. So you can't make the generalization that methylation of lysines automatically is going to lead to gene silencing, because here we've got methylation of lysine that's leading to gene expression. So this is why the histone code, which is right behind my little thing here, probably should move this to the other side if I can. Losing my pointer. It's going to be a combination of the different modifications that basically you have to go in and experimentally check and see what's actually happening as far as these things. And also emphasizes that it's really not so much that chemical modification that happens to the amino acid side chain that's most important. It's actually what it's interacting with, these partners, the histone code readers, that are really the most important. So let's look at a couple of examples of how that works. Here is a general case for chromatin modification. Again, image from your textbook. You have a gene regulatory protein that binds to DNA. Could bind to DNA between nucleosomes. It could be binding to the DNA that's on the nucleosome that's been made available due to this nucleosome breathing process. That gene regulatory protein then interacts with some kind of histone modification enzyme, your writer. So the writer, it's writing the code. So if we're talking about phosphorylation, what kind of protein would that be? That protein would be, man, it is Monday morning, isn't it? So it's going to be phosphorylating serine. What's a protein that phosphorylates something called? A kinase, exactly. What kind of kinase would this be? A histone protein kinase, because what's it doing? It's putting that phosphorylation onto a histone. So protein kinase. So your protein kinase here, that's your writer. Then you have your code reader, which will bind to that modified tail now. And then in many cases, this reader protein will also interact with a very similar writer protein, i.e. your histone protein kinase. So if you have this particular histone that gets modified because of this gene regulatory protein, the fact that you have a code reader protein that interacts with a writer protein means that this modification at this nucleosome is going to lead to the modification at this nucleosome, which is going to lead to the modification at this nucleosome, etc. So this works with kinases. It also works with methylases. And here's a nice example of how we get that heterochromatin formation, the compaction, moving nucleosomes together. Here you have a gene regulatory protein that interacts with a writer, but this writer, instead of being a kinase, is now a methyltransferase. And what it does, it puts methyl groups onto, and in this particular case, lysine 9 of histone H3, K9H3. If you just look for K9H3 in the biomedical literature, you'll find like thousands of references to this particular modification. It's probably one of the best studied of all of them. So this modification interacts with a histone reader complex, which also has a writer complex, which is also a methyltransferase. And what happens is when you have all of these things getting modified, it turns out that these readers and the proteins that are associated with them stay associated with the nucleosomes. It pulls everything together, and that pulling together forms this heterochromatin state, the compacted state, the non-expressed state of your chromatin. Now, hopefully, all of you are going, but Dr. Stedman, you can't have this kind of feedback loop. If you have a positive feedback loop, you're going to turn off all your genes or turn on all your genes. That can't work. So, well, that's actually true. It can't work. And so you have to have a way of dealing with this modification leading to the same modification leading to the same modification. So-called barrier proteins. And so barrier proteins can be proteins that are associated with the nuclear matrix. Remember those matrix association regions I talked about just a second ago? That turns out to be a really good block to this propagation of heterochromatin, or for that matter, and we'll talk about this more when we talk about gene expression, euchromatin, which is the opposite, which is loosening up of your chromatin. So heterochromatin, again, this is your trimethylation of lysine. H3 gets to a barrier protein, and it stops. And this can be just a very physical block, just because these proteins can't associate anymore here, because there's a DNA bound to it. Here it's bound to the nuclear pore, but it could be any part of the nuclear matrix. There are also examples of barrier proteins that literally block the nucleosomes from being modified. These are also cases. And my favorite are these enzymatic activities, which basically reverse that modification that had already happened before. So if you think about a methyltransferase that's putting methyl groups on, then you have a methylase, which will be taking those methyl groups off. And it happens to be bound to a very specific sequence here. So it says you can compact your nucleosomes up to this point, but after this, you won't have it. Question here and then here.
You said any part, the barrier protein can be bound to any part of the nuclear matrix, so it could be bound in the center to like a microtubule filament or something like that?
So it could be bound to, the question is, could it be bound to a microtubular filament or something else which is in the inside of the nucleus? Usually a nuclear matrix is not part of microtubules. There are other parts of the nuclear matrix that we're not going to talk about too much more for the rest of this class. No, microtubules are much more dynamic than most of the rest of the nuclear matrix. But it could be other parts, other proteins which are present in the nucleus. Yeah, so it doesn't have to be the nuclear pore complex. Yeah.
For the example in B, would it skip over those two nucleosomes and continue, or does it end there?
So the question is, would it actually, you be able to propagate this modification beyond those nucleosomes, and just those couple of nucleosomes would be blocked? That doesn't seem to be the case, and the reasoning for that really has to do with the just size of each of these proteins and the modifications that's happening. So if you block the writer complex from writing right here, it can't reach over and write further away. It's just because of the little size of the proteins. If you block a couple of nucleosomes, that's usually enough to block whatever propagation is happening that's actually there. Other questions about nucleosome modification? If he says that, it means yes.
There's nothing that can remove the barrier proteins.
The question is there anything that can remove barrier proteins? And the answer is, of course, yes, because it's a wonderful way to regulate things. And so there are a number of different ways that barrier proteins can be associated or dissociated with the DNA. So you can have modifications here in terms of the nuclear matrix. The binding to DNA you can regulate. If you think about down here, you can regulate the activity of this particular protein. So there are lots of ways of regulating the progression of these feedback loops. Yeah?
This is probably stupid, but in this is a two-dimensional cartoon representation of it, whereas the DNA would be in 3 dimensions, and so you would have them in. How would the barrier protein protect? Does that mean there are just a bunch of barrier proteins everywhere to make sure that it just doesn't jump to another part of the chromatin?
Yeah, this is a very good question, by the way. So this was pointed out here that this is just looking in 2 dimensions. What's really going on in 3 dimensions? As we saw nicely in that animation, where you're looking at where the nucleosomes are relative to each other. Yeah, it's great that you've got a barrier protein that's sitting, go back here, sitting right here and blocking these 2 nucleosomes, but they're really wrapped around. They're around on the outside. And so what about the 3-dimensional kind of interactions here? Do you have multiple barrier proteins, which is one of your questions? How exactly these things work is still actually an open question. It's something that people are trying to figure out. Exactly how these things work. They know that there are barriers that are associated with the nuclear matrix. They know that there are barrier proteins that associate with nucleosomes. We know that there are barrier proteins that are having enzymatic activity. But exactly how they function in the cell is a really open question. It's partly getting back to your question about regulation. Because if you're blocking something from being shut down because it's heterochromatin, then that particular set of genes could be activated potentially by removing what kind of block that was there. And so in fact that is the case. And so it's a very interesting question, and people are still trying to figure out exactly what's going on as far as that's concerned, particularly in the 3-dimensional structure of the nucleus, which we don't understand exactly how all these things are interacting with each other. So that's a great question. Okay, so finally we get to our clicker question. Oh, sorry it took so long. Lower production of mRNA from a gene coded in nucleosomes happens when the nucleosome are in the beads on a string state, like histone H1, contain histone H3 with 3 methyl groups on lysine 9, contain histone H2b with a phosphate group on serine 14, histone H4 with a phosphate group on serine 1. I would not be asking this kind of question on an exam because you wouldn't have open notes, but you guys do, and you've hopefully been listening for the last 10 minutes. Again, feel free to talk about it. It's the idea: interaction, engagement, active learning. It looks like people are going to want more time to do this. Is that correct? Yes. Okay. What we'll do is we'll stop. We'll just restart it, and everyone can vote again. Vote early, vote often. So that last one won't count. We'll start again. Everyone needs another minute. Go. Because there were 108 people who voted last time. Just 50 is probably not right. By the way, clickers always count the last thing that you press. So you can change your mind as many times as you like. Five, 110. Good. Okay, we got more people. So yes, it is lysine 9, histone H3, trimethylation. So obviously I give you enough time everywhere. Someday we'll have 100% on one of these, right? Yes? Unless one of you is really trying to mess with me, which wouldn't, sabotage, always a possibility. Okay. Wanted to spend a couple minutes talking about alternative histones. We mentioned this a couple of times already. Very often it's going to be one of these H2A, H2B modifications here. There are some other examples where you have H3 and H4 that get modified. All of these alternative histones assemble in the nucleosome, so not surprisingly they have that helix with 2 other helixes associated with it, which is the histone fold. But then they've got different tail structures at either end. And it turns out that some of these you find very often in areas of the genome that are very active in terms of transcription, H33, SNPA we'll talk about in just a second, and then also some of the other modifications on H2, DNA repair and recombination, some that overexpressed in HA, and also even an X chromosome inactivation that we'll talk about much later on. Oh, yeah, it reminds me I should have worn my pink hat today, but didn't get around to doing that. So let's look at histone, alternate histone, SNPA. Why SN? CN stands for centromere. It's because you find these histones at centromeric DNA, so right in the middle of the chromosome. This is a part of the chromosome that always stays compacted. It always has all the nucleosomes stuck very closely together. This is what people also call constitutive heterochromatin, so always compacted all the time. Why is that? Well, it's partly because of this SNPA histone which is there, but that also has interactions with the microtubules, like you were talking about, in terms of segregating chromosomes. So centromeres, this is where the whole microtubule structure comes together and then pulls the chromosomes apart. So it's important for the compaction in that part, but probably much more importantly for interacting with all of the proteins that are involved in pulling the different chromosomes apart. So it's not just modifications in terms of transcription, gene regulation, but also in terms of these chromosome structures as well. So these modifications, as well as the alternate histones which can be put in, as we mentioned before, you can get this progression of heterochromatin or euchromatin. Euchromatin is just the opposite of heterochromatin. We'll look at some images of where that is when we talk about gene regulation considerably later on in the course. But the histone modifications that we have here, histone modification, that could be, again, your trimethylation of lysine 9, that then, together with other proteins, will lead to compaction of your chromosome. But other parts, you're not going to have those modifications, probably because there's a barrier of some kind right between these two. And then this is your second part of your genome, which is right next to it. Now, how can you have this heterochromatin that forms, or euchromatin that forms, after you have gene duplication? These things are actually inherited. So I mentioned before, epigenetic inheritance. So this is a non-sequence coded change in your DNA which is still being inherited. How is it being inherited? Well, it's inherited because of those read-writer complexes that we talked about already. So after you have chromosome duplication, you'll have two copies of what you started with here, some of which are going to associate with nucleosomes that have a modification on it. They're going to associate with nucleosomes that don't have the modification on it. And there'll be nucleosomes that don't have modifications on them because they're new nucleosomes. That's new DNA needs new nucleosomes that are formed on it. But if you're right next to where you already had a modification, the read-writer complexes are going to come in and write everything there until you get to this particular barrier. And so after you've had genome replication, as long as you have some of those modified nucleosomes and enough of the appropriate proteins around, the readers and writers, then that will be maintained even after genome replication. And so it turns out that this is exactly what happens all throughout the genome. And this is just shown here as all of these feedback loops. So here's a particular part of the genome that's modified in one way. Here's a part of the genome that's modified in another way, etc. And of course, there are barriers between each of these different pieces here. So chromatin structure is maintained, and it's maintained because of these read-writer complexes which are stimulating that process. So that's what we understand the most about in terms of chromosome structure. So individual nucleosomes, we know down to the atomic level. Nucleosome modifications, we know reasonably well. 30 nanometer filaments, we're kind of trying to wave our hands around. Beyond that, starts to get a lot more wishy-washy. We know a lot less about it, as you probably noticed in my answering a couple of your questions before. A couple things about chromosome organization. This is an overview we'll talk about in a second. Chromosomes actually occupy really specific regions in the nucleus. And this is getting back to that matrix question that we were talking about before. So it turns out that the nucleus isn't just a bag that stuff is floating around in. Each of the chromosomes is a very specific place in that nucleus. And non-expressed genes, heterochromatin, compacted, expressed genes are non-compacted. The way you can see that, if you just look at purified DNA, which we'll look at a couple of examples here, are these in loops that come out from all the rest of your compacted DNA. That's where you have gene expression going on, and the heterochromatin is not. So how do we get from B-form DNA, this structure here, wonderful model from Watson Crick. No one's told me what's wrong with these yet, by the way. You should come down and check them out and tell me what's wrong with models. That gets wrapped up in beads on a string, which we understand pretty well. Got a pretty good idea how the 30 nanometer filament forms. Mostly, again, the side chains interacting here. Probably the tails are really involved. This is now what your different loops. You remember I said about 2,000 of these loop structures. Each of those loop structures is one of those 2,000 you have in the human genome. And then those start to get condensed and even more condensed until you get to a final mitotic chromosome. It's in this step here, going from this condensed form to this condensed form, that we have a couple of proteins that we do know a little bit about. One of those, again, incredibly creatively named condensin, because it's condensing your loop structures and bringing them together to form that metaphase chromosome. Turns out that these are kind of like hair clips. Some of us need to use hair clips, that just hold these individual loops together. They have these long anti-parallel coiled coils. What's a coiled coil? Also known as a leucine zipper. It's these alpha helices that are wrapping around each other. And so basically it gives you a way of getting proteins can be really long in one particular part of the protein. If you can look at these, that's what this is right here. Those are alpha helices wrapped around each other in an electron micrograph here. Cartoon version here. They also have ATP binding, which seems to be important for pulling things together. When you have ATP binding here and actually ATP hydrolysis, it'll pull that hairpin together and basically pull these loops together. Pulling these loops together allows them to all stack up, and that stacking up process gives you a condensed metaphase chromosome. And in fact, if you just look where the condensins are, this is a DNA stain here in blue and a protein stain here in pink. You can see they're all right down the middle of these metaphase chromosomes. And if you do an EM of this, the condensin's right in the middle, and then these are all the loops that are wrapped around it on the outside. We also have another molecule that we know of that has a very similar kind of structure to the condensins. It's called cohesin, just to make things even more confusing. Cohesin is important for holding the two sister chromatids together. So condensin condensing the individual chromosomes, cohesin holding your sister chromatids together. And holding the sister chromatids together, which is allowing them to line up with each other, we'll see really important for recombination a little bit later on in the course. So finish up with a couple of things here in terms of evidence for those looped domains are being the ones where you have actual activity. This is a cartoon version. Here are those individual loop domains. Here's your chromosome scaffold, the matrix we were talking about before. If you look at areas where you have a lot of gene expression, these form these extended loops. So like here's gene expression, etc. And probably the last example we'll talk about today are these what are called puffs. One of the really cool things, anybody looked at these chromosomes, the polytene chromosomes in fruit flies or a number of insects? Get out salivary glands, which are huge in these insects. Look at their chromosomes. Turns out they're all lined up right next to each other, exactly in phase, so multiple copies of the chromosome. And you can see really nice bands on them, except where the genes are being expressed. So it's a big puff. And you can see that because all of these are now, all the dots here, represent the RNA polymerases, and all these bands are where you have all of your genomes lined up with each other. We'll look at chromosome location next time.